Okay, welcome back, guys. USMLE step 1 on first aid 2025. So let's read about T and B cell activation. So I think this is a process that then the T cells get activated first, and then that T cell activate the B cells. So the T and B cell activation, the whole process starts with a antigen presenting cell. So the antigen presenting cell could be even a B cells, a dendritic cells, or Langerhans cells, or a macrophage. So they go present the antigen into the naive or T cell in a whole process to activate the T cells. Two signals are required for T cell activation: B cell activation and class switching too. So the two signals are they describe it down here. So in the process of T cells, specifically T cell activation, the antigen presenting cells ingest and processes antigen. Then this antigen migrate to the draining lymph node. So this is the number one process. The antigen presenting cells ingest and process antigen and then migrate to the draining lymph node to present it, like you see here, to present it into the naive T cell. Then the T cells activation, that's the signal number one. Exogenous antigen is presented to MHC2 and recognizes by TCR T cell receptor on Th cells. This happen in exogenous because in endogenous this is presented to the MHC1. So you can see here exogenous antigens are presented to MHC2. Instead, endogenous are presented to MHC1 on CD8 cells. So you can see here, or 2 or 1 are presented to the T cell receptor. It will be or CD8 or CD4. So it's dependent if it's endogenous or exogenous. So this part here, part 3, there's gonna be proliferation and survival. That's the signal number 2. Remember signal number 1 and signal number 2. So the proliferation and survival co-stimulatory signals via interaction of B7 protein, that is called by CD8 and CD86 on dendritic cells and CD28 on naive T cells. So you can see this here. So this interaction and then the naive T cells CD28 with CD80 and 86 or receptor B7. This whole process, number 4 here, this activate the Th cell and produce cytokines. The T cells able to recognize and kill virus infected cells. So this is whole process is for the naive T cells produce activated naive T cells produce cytokines. Those cytokines, as you can see here in the second part, so those cytokines here they as you can read here, Th cells secrete cytokines that determine immunoglobulin class switching of B cells. So this process here, the number 5, the cytokine activate for the B cells to produce a bunch of immunoglobulins. Okay, so the B cell activation and class switching mean the production of immunoglobulins. So the Th cells activation, as above, I will explain here. The Th cells activation This is a whole process. Then B cells receptor mediated endocytosis. As you can see here, so the antigen is presented to the B cell. This is a mature B cells because the other one was a naive B cells. So this is a mature B cell. Exogenous antigen is presented through the MHC2. You can see here MHC2 and recognized by T cell receptor on Th cell. So it's recognized by T cell receptor on this Th cell. So the CD40 receptor on B cells bind to CD40 ligand, CD4L to the CD40 on the Th cells. This is very high G. I have seen in questions this. They have seen this in questions. I don't know, remember how was the question, but it's asking you about this kind of a receptor. Remember CD4L plus CD40 in a B cell. So the CD40 receptor on B cells bind the CD40 ligand CD40L on Th cell. The Th cells secrete cytokines that determine immunoglobulin class switching on B cell. So these Th cells secrete cytokines that determine immunoglobulin class switching on B cells. Remember we said that the class switching is a whole process through the immunoglobulin are created or formation. So the B cells are activated and produce immunoglobulin M. They undergo class switching and affinity maturation. Let's read what is class switching. So the class switching, or also known as isotype switching or class switching recombination, is a process where B cells change the type of antibody they produce without altering their ability to recognize a specific antigen. This switching involves altering the constant region of the immunoglobulin heavy change, which determine the antibody functional properties, such as its ability to neutralize pathogens and activate complement. So the purpose of this is the B cells initially express immunoglobulin M and immunoglobulin G antibodies. When they find an antigen and receiving appropriate signals, the B cells can switch to produce other antibodies class like immunoglobulin G, immunoglobulin A, or immunoglobulin E. This switching enhances the antibody ability to combat different type of pathogens or immune responses. This is important here. If there's a defect in the class switching, you can have a disease called hyperimmunoglobulin syndrome. Hyperimmunoglobulin syndrome, where B cells are unable to switch to another antibody class besides immunoglobulin M, potentially compromising immune function. So it's like the immunoglobulin M is the basic one. So from that one, all the different immunoglobulins are transformed. So if you have the ability, or if you have the deficiency of this enzyme, that is the one who regulate this whole process, or if you have a class switching impaired, you're gonna have the disease called hyperimmunoglobulin M syndrome. So the hyperimmunoglobulin M syndrome are a group of rare, very rare primary immunodeficiency disorders characterized by the body's inability to switch from producing immunoglobulin M antibodies to others anti-immunoglobulin types like immunoglobulin G, immunoglobulin A, or immunoglobulin E. So the most amount that's going to be is only going to be immunoglobulin M. So all the other immunoglobulins come from immunoglobulin M. So won't be any other kind of immunoglobulin, only immunoglobulin M. So this results in elevated levels of immunoglobulin M and a deficiency in other antibody types. So inability to switch antibody types, this is the producer's bias. So the most defining features of this defect is a class switching problem, preventing the production of sufficient immunoglobulin G and A or E antibodies. So there's going to be elevated immunoglobulin M levels, while the immunoglobulin L levels are either normal or elevated. The immunoglobulin G, A, or E levels are absent or significant decrease. So the patients with the immunoglobulin problems, like this case, hyperimmunoglobulin, they're gonna have symptoms, are prone to infection. They're gonna have very risky to have infection, particularly those caused by bacteria, virus, and fungal. Other potential symptoms are dependent on a specific type. I think this part here is important because it tells you about two different types of immunoglobulin, hyperimmunoglobulin syndrome. So type of immunoglobulin syndromes are X-linked hyperimmunoglobulin M syndrome. This is type 1, and autosomal recessive hyperimmunoglobulin syndrome. So if you can remember the receptor that we talked about before, CD40L on CD40 and CD40, when they are lack, let's see, X-linked hyperimmunoglobulin M syndrome. This is the most common type, caused by mutation of the CD40L G gene. So this gene is the one who codes for the receptor that we just talked about, CD40L, CD40 ligand. So autosomal recessive hyperimmunoglobulin syndrome, this type can be further classified into the several subgroups which depend on the gene that is affected by the sample, in this case CD40 and NCD40. So we can see here, CD40L or CD40 ligand, this give you one kind of immunoglobulin, hyperimmunoglobulin M disorder. So the X-linked immunoglobulin M disorder, as we can remember here. So the next concept is called energy. The concept of energy is a state during which cells cannot become activated but exposed to its antigen. The T and B cells become energetic when exposed to the antigens without co-stimulatory signal. So another example of peripheral tolerance mechanism. So this concept of energy, I think, is best expressed here. So self-tolerance in the context of the immune system refers to the ability to distinguish between self and non-self foreign invaders like pathogens or toxins. So self, like the body's own tissues and cells, and non-self, like foreign invaders like pathogens and toxins. This is crucial in preventing the immune system from attacking its own tissue, a condition known as immunity. Energy-related aspects to self-tolerance involve the immune system ability to maintain a stable and functional state, while also efficient responding to the threat without causing harm to the body. So self-tolerance in the context of the immune system, particularly in the USMLE, refer the ability of the immune system to differentiate between self-antigen and foreign antigen, preventing it from attacking the body's own cells. This is crucial for preventing autoimmune disorders when the immune system mistakenly attacks healthy tissues. Okay, so let's read now about antibody structure and function. This is an antibody. It's formed that more likely to structure this is the heavy change and light change. They have a light change here, this part here, and a heavy change, combination of heavy change and light change. So this region here is the FAB region. This region down here is the FC region. This region is very important here because where's the complement bind and the macrophages binding too. So let's read the antibody structure and function. So the FAB, the fat fragment, consisting of light and heavy chains recognize antigen. So the FAB region is this one here, is formed by the heavy part and the light part, the light change. So the heavy chain and the light chain, this portion here, as we said, is the portion forming the FAB region. This one is the one who recognizes antigens. There are antigen binding here in this part here. So the FC region of the immunoglobulin M and G fix the complement is this part here, the FC region here, down here. That's the part where the fix the complement on the macrophages bind there too. So the FC region of the immunoglobulin M and immunoglobulin G fix the complement. The heavy chains contribute to the FC and FAB region at the same time. The heavy one contributes to the FAB region and for the FC region at the same time. The light chains contribute only for the FAB region. So this is the light one. This is only contribute for the FAB region. In the graphic here, you can see that part. So the FAB region, remember, it's good to remember because given this mnemonic here, they have fragment, antigen binding, fragment, antigen binding, fragment, antigen binding, fragment, antigen binding. This is the FAB area here, FAB area, fragment, antigen binding, this part here. So this determines isotypes, unique antigen binding pocket, only one antigenic specificity expressed per V cell, only one antigenic specificity expressed by T cells. So remember the FAB region, this region here. This is the fragment antigen binding, this area here. It determine the isotype is the unique antigen binding pocket, is the unique place where the antigen is gonna bind. It's only one antigenic specific expression for B cell. So the other part, the FC part, we have here the 5 C's. It's constant. It's form of carboxyl termination. You can see here CH carboxyl termination is the complement binding place, complement binding, carbohydrate site change, and converse. That means that determine what type of immunoglobulin. That means that confers the isotype. Could be immunoglobulin M and immunoglobulin G. So depending on this region, you're gonna this is gonna turn into immunoglobulin B or D, M or D. It's where the generation of antibody diversity, antigen independent, generation of antibody diversity. Remember, generation of antibody diversity. So this process is this is a process that contains the random recombination, random addition of nucleosides, and random combination of heavy change. So the random recombinations on the VD, BDJ heavy change genes by the activation of the RAG G1 and RAG G2. So there RAG1 and RAG2 are essential protein involved in a VDJ recombination, a process that rearranges antigen receptor gene in developing B and T cells. They form a complex that initiates DNA cleavage and specific sites called recombination signal sequences. This process is crucial for generating a diverse range of antigen receptors and ensure proper immune function. So remember, random recombination of BJ in light change or BDJ in heavy change. Those genes are that random recombination is made by RAG1 and RAG2. We just read what is this about. So the random addition of nucleotides, the random addition of nucleotides to the DNA during the recombination by terminal deoxynucleotidyl transferase, the TDT. So the TDT, or terminal deoxynucleotidyl transferase, is an enzyme crucial for generating diversity in the immune system, specifically during the lymphocyte development. In the context of USMLE Step 1, understanding the TDT is important, particularly related T-cell receptor and immunoglobulin diversity. So the TDT adds non-template nucleotides between V and J gene segment during the VDJ recombination in both T-cell receptors and immunoglobulins. This end region adding increases the diversity of TCRs and LGS, which is essential in recombination of wide range of antigens. Some concept to remember: TCRs are T-cell receptor and found on the surface of the T cells and are involved in recognition of antigen presenting cells. IgS or immunoglobulins, also known as antibodies, are produced by B cells and are involved in the recognition and neutralization and pathogens. The BDJ recombination, this process rearranges gene segment by the BD and J. These create unique receptor on the surface T cells and B cells. So remember, random addition of nucleotides by the TDT. So we have to random combination of heavy change with light change. So we're gonna have generation of antibody specificity, antigen dependent. So through the whole process, we're gonna have generation of antibody specificity, but antigen dependent. So there's gonna be somatic hypermutations and affinity mutation. That's going to be in the variable region, and then isotype switching in the constant region. Remember, constant region, isotype switching. A variable region is going to be somatic hypermutations and affinity mutation. So next, let's talk about immunoglobulin isotypes. The different type of immunoglobulins, which are the most important, which one is the most common, which one is the most frequent, which one is the first one to arrive, and a specific what each one do. So immunoglobulin isotypes. So all isotypes can exist as monomers. Mature naive B cells pre of the activation express immunoglobulin M and immunoglobulin D. Remember, doctors, MD. This is the first one. So mature and naive B cells pre of the activation express immunoglobulin M and D. So the cells even mature or not new B cells, they present in their surface immunoglobulin M and D. Remember, doctors MD. So those one can differentiate in the germinal centers. Remember the germinal centers, the B cells, that's the place of the germinal cells live. This is the place where the B cells live, the germinal centers. Okay, remember that, germinal centers. So they can differentiate in germinal centers of lymph node by isotype switching, doing the isotype switching. This generates rearrangement induced by cytokines CD40L, remember, ligand, CD40 ligand. If there's deficiency in this CD40 receptor, remember, we're gonna have hyper immunoglobulin M. Remember that. You guys have to remember that, repeated several times. Hyperimmunoglobulin M syndrome is for deficiencies of CD40L, one of them. So they might differentiate in germinal centers of lymph node by isotype switching. This is the gene rearrangement induced by the cytokines and CD40L into the plasma cells that secrete immunoglobulin A, G, or E. For B cells, an immunoglobulin M, it would say, is the mom. So immunoglobulin M, let's say that is the mom, and immunoglobulin G is the dad. Of those different kinds of immunoglobulin, and they later gonna be age. So mom and dad, they are the mom and the dad of those all immunoglobulin, and then in the plasma cells they're gonna age. So immunoglobulin A, G, and E. This is a good mnemonic to remember that the first two immunoglobulin are MD, like a doctor, mom and dad, and they to the plasma they were gonna age. So let's read that again. For B cells, immunoglobulin M is the mom and immunoglobulin D is the dad, mature to plasma cells as they age. Okay, very good mnemonic. So affinity refer to the individual antibody antigen interaction, while avidity describe the cumulative binding stretch and all antibody antigen interaction in a multivalent molecule. So we have to read again the concept of affinity and avidity. Avidity is like a more specific, affinity is more specific for an individual antibody, this is specifically, and the other one is more general. We have to read those concepts again. Affinity and avidity. Let's see affinity. So in immunology, affinity refers to the strength of the binding between the strength, or how strong is the binding between a single antibody binding site, paratope, and a single antigen binding site, epitope. It's the force of the single antibody antigen bond. Affinity is crucial for the immune system ability to recognize and neutralize antigens. Affinity mutation, a process where antibodies develop high affinity for antigen over time, is also a key concept. The aspect of affinity in immunology. And this is the other concept, avidity, but the definition is almost the same. But it looks like the avidity is part of the affinity. So let's see, immunology, the avidity refers to the total strength of binding between an antibody and its antigen when multiple binding sites are involved. It is essentially the cumulative effect of the individual affinities of all binding interactions. High avidity indicates a strong overall binding, while low avidity suggests a weaker interaction. Factors influencing avidity. So in between the factors influencing avidity, we have then the affinity and the valency and the structural arrangement. So affinity, the strength of individual antibody antigen interaction. That's the affinity. The valencia or valency is the number of binding site of both the antigen and the antibody and the antigen. And the structural arrangement, the spatial arrangement of binding site of both molecules. So as the book describes, we have the affinity refer to the individual antibody antigen interaction, this say individual, while avidity is described the cumulative, like a more than one binding strength of all antibodies antigen interaction in a multivalent molecule. Okay, affinity just for one, avidity for more than one. Think about the ability of that person have bouncing a lot of objects, so different kind of objects in the air. That's the ability. Okay, so we have here immunoglobulin G, A, M, D, and E. Remember, M and D are the mom and dad of all those immunoglobulin. So let's read about immunoglobulin G, the main antibody in a second response to an antigen. The most abundant isotype in serum, immunoglobulin G. Remember, this one is the one who creates the baby, because this is the only one who goes through the placenta. So the main antibody in second response to an antigen, most abundant isotype in serum, fixed complement, it's opsonized bacteria, neutralized bacterial toxins and virus, is the only isotype that closes the placenta. This is important for steps. Is the only isotype that grows the placenta, provides infant with a passive immunity that starts to wave after the birth. So this immunity disappears 6 months after the birth. The immunoglobulin G greets the growing baby, associated with warm immunity, hemolytic anemia. Warm weather is good. Warm weather is good to remember, because the warm weather is good. The G on good is related to warm weather, warm, so it's associated with warm immunity, hemolytic anemia. Associated with warm immunity, hemolytic anemia. Immunoglobulin G is the main antibody in second response to an antigen. Most abundant isotype in serum, the most abundant. Fixed complement, opsonized bacteria, neutralized bacterial toxin, and virus. Remember, it's the only one who crosses the placenta and is related with warm immunity, hemolytic anemia. So let's jump into immunoglobulin A. Immunoglobulin A. So the immunoglobulin A prevent attachment of bacterial and virus to mucous membrane. Does not fix complement. It's a monomer. You can see here have two parts. And dimer with a J change when secreted. So it's a monomer, just one when it's in the circulation, but when it's secreted, most of the time in the mucosas, it's become with the J change in two parts on dimer. So it crosses epithelial cells by transcytosis. It crosses epithelial cells or membranes by transcytosis. Is produced in a GI tract system. GI. It's by example by the Peyer plates. Peyer plates. Protect against good infection. By example, Giardia. Most produced antibody overall is the most produced antibody overall, but has lower serum concentration. Is released into the secretions like saliva, mucus, tears, and it is very abundant in milk. Breast milk is very abundant in breast milk. Picks up secretions components from epithelial cells, which protects the Fc portion from luminal proteases. That was immunoglobulin A. Remember, is very frequent seen in mucosal. Protects us. It protects us from Giardia and all that kind of mucosal affections. It is very common in the breast milk. Doesn't cross the placenta, but is common in the breast milk. So let's see the immunoglobulin M. Immunoglobulin M, the first antibody to be produced during an immune response. Fixed complement, antigen receptor on the surface of B cells, is a monomer on a B cells, pentamer with the J joint when secreted. Pentamer means that's formed by 5 different type of structure. You can see 5 structure all together by the J joint. So compared with the immunoglobulin A, that is only 2 monomer when it's secreted. This is pentamer when it's secreted. So it's monomer on B cells and pentamer with J when secreted. Is a pentamer enables avid binding to antigen, while humour response in both. It is associated with cold immune hemolytic anemia. Cold immune hemolytic anemia. Remember, the immunoglobulin G is warm immune hemolytic anemia. Warm. Remember, warm weather is good. So this one is associated with cold immune hemolytic anemia. So one thing to remember is that this look here in pentamer shape, it looks like a fan, like one of those fan when it's hot weather. You use one of those fan to get a little because it's too hot. So you use a fan to get cold. So that's why this one, immunoglobulin M, is associated with cold immune hemolytic anemia. Cold immune hemolytic anemia. The pentamer shape makes you remember as a fan. Fan is associated to refresh yourself. So the immunoglobulin D expressed on the surface of mature naive B cells. Remember, MD, mom and dad, as this one too, as immunoglobulin M. The immunoglobulin D is expressed on the surface of the mature native B cells. There's normally low levels are detected in the serum. So immunoglobulin D, it looks like the immunoglobulin D doesn't do that much. That's like a mom and dad, that the dad never do anything, the mom always do everything. So that's the way I remember. That the mom is the one who do everything. The dad doesn't do almost anything. And the one that everybody know is the immunoglobulin E. That's a very common, recognized for everybody. The immunoglobulin E. So bind the immunoglobulin E bind mast cells and basophils. They produce a cross-link when exposed to an allergen, mediating immediate type 1 hypersensitivity through the release of the inflammatory mediators such as histamine. Contributes to the immunity to parasites by activating eosinophils. So it's related when there's an infection on the parasital in GI intestine, when there's like a parasite infection, they activate eosinophils too. So contribute to the immunity to parasites by activating eosinophils. So immunoglobulin E bind mast cells and basophil. They produce a cross-link when exposed to a large allergen, mediating immediate type 1 hypersensitivity. Immunoglobulin D expressed on the surface of mature and naive B cells. Normally low levels are detected in the serum. Remember, immunoglobulin M is a pentamer form. Pentamer enabled avoid binding and change while humoral response involved. Is associated with a cold immune hemolytic anemia. This is a immunoglobulin M. Remember the immunoglobulin A is more in mucosa, is produced in GI tract, in the inferior plexus, and protects against gut infection like Giardia. Most produce antibody overall, has lower serum concentration, released into the secretion, tears, saliva, and mucus, is present in the breast milk. And the most important to me, I think so, is immunoglobulin G, is because it's across the placenta and is associated with a warm immunity hemolytic anemia. Okay, in the next video, we're going to talk about complement, activation, pathway, and function. Complement, and we're going to keep reading about complement disorders, specifically C1, C4. We're going to keep C1 inhibitor deficiencies in very high year. This is another pathology, very high year, paroxysmal nocturnal hemoglobinuria. We're going to read about the different interleukins, 1, 6, 8, 12. And we're going to talk about respiratory burst. This is a process through the bacteria are destroyed in phagocytosis. Cell surface protein. Thank you very much. See you next time. Good luck studying. Good luck with your test.